Let me introduce Sina Trautmann-Lengsfeld a little bit biographically. She was born in Bremen and studied psychology and focused on the neuroscience of emotion perception during her PhD at the University of Bremen. In 2010, she started her first postdoc at Professor Christoph Herrmann's lab in Oldenburg, where she explored the neural correlates of social conformity with electroencephalography, the EEG. In the end of 2011, she started her second postdoc at Professor Andreas Engel's Department of Neurophysiology and Pathophysiology at the University Medical Center Hamburg-Eppendorf. Here, her main scientific interest became the investigation of the neural correlates of synesthesia, especially grapheme color and sound color synesthesia, during multisensory processing examined with magnetoencephalography. I hope I pronounced this right. Magnetoencephalography, MEG. And today, Sina will present us her talk, which is titled Multisensory Processing in Synesthesia. Thank you for coming.
Thank you, Regina, and I'm very glad to be here. I feel quite honored to be part of the great speaker list, you know, all the interdisciplinary speakers from art, science. Very, very nice. Great job you did there. Can you hear me? Yeah? Like this? Okay. So we're going to switch topics completely, and I hope that I can give you a nice overview of the work we do. It's neuroscience, so it might be a little more dry than what we've heard before, but I hope that I can, you know, catch your attention and you stay with me. I'm going to try to break it down so that you can really understand what I mean. This is my agenda. I'm going to talk about unisensory experiments. Well, first of all, I have to tell you a little bit how we as scientists try to define synesthesia, because as we've already heard, synesthesia is very complex and very individual, but we have to find a common place how we can kind of classify the different forms, because we always need very standardized settings in our experiments. So I'm going to talk about that a little bit. Then I'm going to tell you a little bit about different models in neuroscience. We've already heard a little summary from over there earlier on. Then we're going to talk about unisensory processing in synesthesia, although this unisensory processing is still multisensory, kind of, because, you know, one sense is activated and another one is activated simultaneously in synesthesia. Then we go on to multisensory integration, which is a rather rarely studied topic until now in synesthesia, mostly independent of synesthetic experiences. That's my, that's the topic of my work, actually. And I'm going to tell you a little bit about my experiments and the first results, but not brain results, just behavioral results, but still. Then I'm going to give a little outlook. Let me start with some famous people who have synesthesia, among others who are sitting right here. There are people like Kandinsky, we've heard about him before, Eugene Bleuler, Wittgenstein, Daniel Tammet from England. He is a savant as well, and he has color and digit synesthesia. Jimi Hendrix, Stevie Wonder, the singer from Marina and the Diamonds, Tori Amos, they all have colored sound color synesthesia, and they also use that for their work. So that's just a small overview, and the list is of course never complete. Okay, how do we define synesthesia in neuroscience? For us, synesthesia is actually an involuntary and automatic phenomenon. It always happens, you know, like when I talk, colored hearing synesthetes, they see the color immediately. They cannot suppress it or inhibit it. It is highly consistent, so if you ask a synesthete today, what is your color for A, for example, it will be red, for example, today. It will be red in a few years as well. It hardly changes. It begins usually in early childhood, although we know very little about it, and Professor Emmerich has already talked about the possibility that synesthesia, that we are all synesthetes when we are babies, and it just prunes away when we grow up, and these connections in synesthetes just stay. This is something that we really have to see in the future. It runs in families. often, so it's quite interesting to also look at the genetic perspective. And yeah, the prevalence is not consistent at all. It depends on who you ask and how the different scientists are classifying synesthetes. It goes from 1 to 25,000 or to 1 to 2,000. Synesthesia is always, when we talk about synesthesia, there's always an inducer, like for example a letter or a digit or a sound, and a concurrent synesthetic experience, which is actually the activation of another sense that non-synesthetes would not experience. So the stimulation of one sensory modality leads to another sensory modality that is activated, and that's so interesting. Over here you see 3 examples for letter color synesthesia from one person. The alphabet could look like this. This is an example for also digit color synesthesia, which is kind of localized in space. And this is where actually the different months of the year have a certain color and are spatially distributed over a year. There are many, many types of synesthesia, and this is just a small part of a small list of synesthesias that have been reported before. And like I said, you always see the inducer first and then the concurrent synesthetic experience. So there's, of course, the grapheme color, then the musical or sound color synesthesia, or also odor color, taste color. There's orgasm color, there's touch color, but there's also sound and touch, for example, that we've heard before, or taste and sound and taste. So many, many different ones. For my research, we had to focus on 2 different forms, and those are the forms that are most, that you find most often. And those are grapheme color synesthesia, including also weekday and month color, and auditory or sound and color. I also want to show you 3 examples of synesthetes. The first one is from Jamie Watt. Jamie Watt is a very famous professor from Brighton who has studied synesthesia quite extensively. And he published a review, and he showed an example of a grapheme color synesthete who also has the weekdays in a certain sequence and also in a certain color. This is actually from one of the synesthetes that I was allowed to measure recently. I asked him to draw his name, or to paint his name. His name is Wolfgang. I'm allowed to say that. And this is how his name would look like. It goes from left to right, and it has black, orange, and blue colors going from one side to the other. And this is just a pure piano tone. Unfortunately, I forgot which one it was. And then there's another quite interesting example. I think it's about a synesthete who said, I quote, "I avoid middle C when I play the piano because it's an earthy, musky smell I don't like." It's kind of what you told before, that, I don't like the taste of, of your name. It's similar to that. Okay. Now, I said we have to classify synesthetes, and it's very difficult because it's complex and it's individual, and especially with sound, color synesthetes, there are always pitfalls in these classification tools that we have, but we have to use them, to kind of standardize our subject group or our sample. And there are many different ones. One of the first ones was probably the test of genuineness by Simon Baron-Cohen, also a very famous synesthetic researcher. Then there's the color synesthesia test from the Netherlands. There's another one from Düsseldorf. And also by Janina Neufeld and Christopher Zinke, they did a very extensive, half-standardized interview that I used myself as well to get a better overview of the synesthesia that my subjects have. And then there is also a different one. There's a questionnaire that tries to separate the associators from the projector synesthetes. The associators synesthetes are rather the ones who see who kind of associate the color to a grapheme, for example. Yeah, they see it in their mind's eye, but it's not projected onto the screen or onto what they read. That would be the projector synesthetes. And we try to differentiate it, and there are hints that projector synesthesia is differently processed in the brain than associators. Projectors is what I think, you're probably also a projector synesthete because you project. Now you're associated? Okay, you see it in your mind's eye. Okay, the project is really projected on a piece of paper. What we use, or what I basically use, is an online test battery. It's synesthete.org. Some of you might know that. And there you can actually test yourself and see whether you're a synesthete as well. I'm going to give you a short introduction how that looks like. And it's been used very much, and it's quite reliable, but it has its pitfalls, like I said before. Like, for example, for grapheme color synesthetes, a test would look like this. You always have two pieces. The first one is the classification of the grapheme to the color. You have a little square with different colors in there. You can change the color from blue to red to yellow here on this scale, and from very bright to dark on this scale. Here you have the letter that you are supposed to classify, and this is the color that you will choose. If you have chosen the correct one, you can go to submit color. You do that for every single grapheme 3 times in a randomized order. That's the first thing, and then you get a consistency score to see whether the colors that you've chose are consistent over the 3 times that you're asked for this grapheme. Then, and that is very important because there's been research which showed that people can memorize colors with letters. You know, I mean, we have 26 letters, and some of them might have a good memory. Then you can memorize the color. But no, there's an additional test that can kind of exclude that it's just a memory thing, and that's the speed congruency test, where you see the grapheme in your color or in another color for 1 second, and then you're supposed to say yes, it matches or it doesn't match. And you have to have a high consistency score in this test in order to be actually classified as synesthete. They have that for sounds as well, where you can choose the color. One of the pitfalls here is that they actually show the note here. We have worked on a program where the note is not shown, where you just hear the sound, and then you're supposed to, associate the color. And the same with this, you also, of course, do the speed congruency test. Those are just 2 tests in this online test battery. Results would look like this. On the left, you see the result sheet of a synesthete. On the right, you see a result sheet of a non-synesthete. And you can see here, I hope you can see it, N, for example, has always been green for this synesthete, yeah? Or Z always like a green, yellowish kind, or K pink. So it's very consistent. So if the score here is below 1, you're classified as synesthete. And also here, the speed congruency test, you have to have an accuracy above 85%. So this person is definitely a synesthete. On the right, you see a non-synesthete. You can see that, for example, the 9 has 3 different colors. Yeah? Or even though the A looks like green in all of the 3 letters, the other letters are all kind of mixed up or have no color. So also with this score, it's 4.1. It's quite easy to say no synesthete. So if you're interested and you want to do it yourself, you can go ahead, and I would be glad, because I'm still looking for participants for my study, if you just put my email address in there so I could look at your data and then maybe invite you if you're interested in participating. I also have some handouts later on if you're interested, just ask me. Okay, so back to the definition. The question is, why is it so interesting for us, yeah? For neuroscientists? Well, the thing is, and Professor Emmerich has already talked about that before, that these senses are somehow connected. So it's kind of a multisensory experience, and multisensory experience has been quite a lot studied in non-synesthetes before. So we know a little bit about how it works in non-synesthetes, but maybe synesthesia can give us hints about, even more hints to understand how the brain works when it combines 2 senses. For example, hearing, seeing, or taste, or smell and seeing, stuff like that. Because senses always are enhancing each other somehow, or not always enhancing, sometimes also decreasing each other. So especially for us, we want to know, because there are still some doubts how synesthesia is actually processed. How does it work in the brain? How is it processed? And how does especially multisensory integration in synesthesia work? So I'm going to give you a little overview of first unisensory, unimodal experiments, and then I will go on to the multisensory part. But before that, I want to talk about two main theories actually in synesthesia. And that's, for one, the disinhibited feedback theory by Grossenbacher and Lovelace. And this theory says that we process information in the brain and that it is, in synesthets, it is converged in the multisensory nexus. Let's call it like that. The multisensory nexus is a part in the brain that combines experiences, perceptions from different modalities, etc. So this is here. We have a stimulus that is processed from the back, for example, visual stimulus from the back to the front. And in synesthesia, these feed-forward processes go back again. And the back way is disinhibited, which is not there in non-synesthetes. That means we don't have additional connections. We just have a feed-forward and feedback connections that lead, if we go up here, it goes back to both sides to the concurrent synesthetic perception. Is that understandable? Yeah? Okay. There's another theory that is based on actually additional connections, because during brain development, our brain areas are connecting, and when we grow up and we become adults, these connections decrease if we don't need them. They just prune away, they die. And there's a theory in the cross-activation theory that this doesn't happen in synesthetes. They keep those nice interconnections between the different areas. For example, if we see a grapheme, it doesn't go up to the multisensory nexus and then go back. No, it goes immediately to the color area, to the grapheme area, and then over to the color area. And that's why we have these simultaneous perceptions. I want to put that in a nutshell so that you can have a brain in front of you, yeah? So this is the brain from the side, from the right side. We have the front here, we have the back here. This is where we process seeing, this is where we process hearing, and this is the multisensory nexus. And first of all, the cross-activation theory could look like that, yeah? We get information to the color and grapheme area, and they are just interconnected. This is displayed with the arrows here. Or if we have auditory sound color synesthesia, it could go to the auditory cortex first and then go back to the color area like that. Yeah? This is the cross-activation theory. The disinhibited feedback theory, displayed in blue, would go like this. We would see a grapheme that is processed in the grapheme area, which is here, and then it would be processed in the multisensory nexus, but then there are feedback connections to the color area, and that's why the color would be simultaneously activated. Okay, let's keep that in mind, because we're going to look at brain data later. So, one of the unisensory experiments were about, for example, what can we learn from synesthesia? Is there any win if we have synesthesia? And yes, there is. In some experiments, there is actually. If we show people, non-synesthetes, a screen like this, you know, with different letters, and the task is to see, is there the letter E in there? We have to kind of search first, yeah, as non-synesthetes. As a synesthete, maybe my E is red, and the other letters have different colors, so they kind of come out immediately. So what happens is that synesthetes are much faster than non-synesthetes. You can see that in the graph here. They measured 6 different grapheme color synesthetes. The gray bars are the synesthetes, the white bars are the non-synesthetes, and the black bars are controls that saw not this black and white display, but the color display. And of course, if you see the color display, even the non-synesthetes can say immediately, hey, there's an E, so I'm fast. So they were the fastest and the most accurate ones. Synesthetes were almost as, well, especially this one, was almost as good as the non-synesthetes with the color. So yes, there is definitely a win situation in some experiments, especially here. We have a similar one here, but that one shows actually that synesthesia can also kind of impair your performance. They showed screens in the color of different graphemes, for example, this color was congruent with the color, well, no, I'm going to start again. The task was to say, is there a 4 or a 2 in this bunch of numbers? And the bunch of numbers were presented on a screen colored either congruent, in this case, or incongruent, in this case, to the grapheme color of the synesthete. So in this case, the synesthete would not find the 4 that quickly because it has the same color as the background, right? In this screen, it would be much easier because the background color is different from the color from the actually searched number. And you can see that here in the reaction times as well. Synesthetes are worse, slower actually, in the congruent condition than in the incongruent condition. So it can also, synesthesia can also keep you from being fast. Okay, then there's the Stroop test, which is also, maybe you know that, it's kind of a famous psychological test. The task is to say the color of the color words. So, for example, here you're supposed to say green, here you're supposed to say orange, here you're supposed to say red. It's quite easy because the color matches the color that you read. But as soon as it becomes incongruent, for example here, the color word red is presented actually in green, it becomes much more difficult. Maybe you can try it on yourself real quick and see. It is more difficult, right? So what we see as psychologists, you make more mistakes because you are under a lot of pressure when you do this. You make more mistakes and you become much more slower. You can do that as a synesthetic experiment as well. Okay. Before I go to that, if you compare synesthetes, which are presented here in black, and matched controls in white, you hardly see a difference in reaction time, yeah, between both groups. However, if you do the same task with graphemes that are either congruent with a color, like for example if you have a synesthete where R is always green, when you present it in red, this impairs the reaction time. And you can see that here. For non-synesthetes, it doesn't matter in which color the letter is presented. They're always fast in the congruent and incongruent condition. But for the synesthetes, they become much slower when the color is not matched to their own grapheme color. So the question is, what does that tell us about the different theories? The thing is, we cannot really answer it yet. We need to look into the brain. And how do we do that? I'm going to give you a short overview because I'm not sure whether you're familiar with all the different methods. There are basically 2 methods. Well, actually there's PET as well, but I'm going to skip that. We're just going to talk about functional magnetic resonance imaging, abbreviated with fMRI. We'll talk about MEG, magnetoencephalography, and electrophysiology. fMRI, maybe you know that. It's a magnet, and it can measure the bolt response, well, the amount of oxygen in your brain. And the advantage of this method is that you can very precisely locate the areas that are activated, very precisely. The pitfall is that you have a very low temporal resolution. That means if you find activity, those are, for example, brains from below, and you see these blobs on there, right? That means the brain is activated somewhere. You can say where, but you cannot say how do they interact on a temporal basis. So therefore, you need, as a complement, MEG or EEG. They're based on the same process. Here, this is rather an indirect measure of brain activity because it's just, the blood flow. Where the brain is activated, it needs more oxygen. It makes sense. But it's indirect because You don't really measure cell activity per se. In EEG, you measure cell activity, the neurons that are activated, neuron assemblies. And you have a very high temporal resolution in milliseconds. So that's the advantage of this method. But you have a lower spatial resolution. While you have it here in millimeters, you have it here only in centimeters. And to give you a little hint how this could look like, what you do here is you just average activity and you see graphs like this. You don't really have to understand them. I'm going to guide you through them later. Okay, so these are the methods. We need them to see whether, like for example, these areas are active simultaneously, one after another, or how does that actually work? So fMRI we need for localization for the different areas, and MEG and EEG we need to say, okay, when is what active and how do they sync to each other? There was once a very nice study from Hubbard and Ramachandran in 2005, and they actually showed graphemes in black or in white on a black screen, and synesthetes were looking at that, and they saw what kind of areas were activated. And what they could show nicely in synesthetes in comparison to controls, you have in pink, you have the color area again, you know, the area that is activated with color. And in blue, you have the grapheme area that processes letters. And what you see here is over the color area, you see these orange blobs, right? And that means that synesthetes have activation in the color area, even though color is not presented. So there seems to be an interaction between the grapheme and the color area. Controls don't have that. They have activation here in the grapheme area, but not here in the color area. The pink blob doesn't mean there's activation, it just means there would be the color area. So there's no activation. But now the question is, how does that happen? Is that via the multisensory nexus, or is it simultaneously? And this is quite cool because a PhD, now postdoc from Ramachandran, did an MEG study with a similar setup. And what they could show is, they also tested synesthetes and controls again. And here you see a graph for the color area, and here you see the graph for the grapheme area. For the grapheme area, you see red is the controls, and blue is the synesthetes again. You don't see a difference, do you? Well, here a little bit, but I mean, it's not that obvious. So they both kind of process graphemes. That makes sense, right? But when you look at the color area over time, and this is in after only 100 milliseconds, you can see that the synesthetes have much higher activation here. So they kind of do activate the color area simultaneously with here the grapheme area. So that is a proof for the cross-activation theory that it might happen simultaneously somehow. I'm going to skip that. This is actually a study from Janina Neufeld and Christopher Sinke that Professor Emrich was talking about before, and I'm very glad that he didn't go into detail because now I can. It was an fMRI study, and they had auditory visual synesthetes, so not grapheme color synesthetes, auditory visual synesthetes, and they presented tones and chords and saw what kind of activation happens in the brain. And what they found was that synesthetes, in comparison to non-synesthetes, hence controls, had activation only in this multisensory nexus. So this would be evidence, actually, for the disinhibited feedback theory that this multisensory nexus has to be activated to actually have the color experience. Then there's another study, an EEG study, also with auditory visual synesthetes. They did not find any differences between synesthetes and controls. So you see that there's a wide difference actually between studies or between the results that we can find. It's quite contradictory, and we cannot always say it's always like that. There's still a lot of research to be done. Another study I want to talk about is, I don't know whether you have heard about the method TMS. It's transcranial magnetic stimulation. You put a magnetic field on a different region of the brain, and you can either enhance that region, make it more active, or inhibit it, make it less active. And this is what Esterman and colleagues did. They also showed, actually this is the Stroop test, the synesthetic Stroop test that I was talking before about. They had congruent colors with the graphemes and incongruent ones and neutral ones. But let's focus on the incongruent ones. This is where we would expect the synesthetes to make more mistakes and be slower. And what they did is they stimulated the multisensory nexus and inhibited it, so it was out of order. Yeah? And what they found is that this impairment in reaction time and in error rate decreased. So all of a sudden, this interference that they had before without the stimulation was gone. And this is also an indirect, or actually it's a causal proof, or it might be a causal proof that synesthesia for the disinhibited feedback theory. It might be. It still has to be replicated, of course. Okay, so what you see is that there are some studies showing V4 activation, and cross-activation, and there are other studies that show actually in the multisensory nexus an activation. So what might be the difference between these studies? One of the differences might be all of these studies have not differentiated between projector and associator synesthetes. And Tessa van Leusen, she's now in Frankfurt, and she did a very nice study because she separated these two groups and looked at maybe there are different ways how these synesthetes are processing graphemes. And she did an fMRI study, and she set up a model. So this model is similar to what you saw before about the different theories, right? We have here the multisensory nexus, we have the color area, and we have the grapheme area. And what she found is, and that's quite cool, I think, for the projector associates, she found a concurrent, well, actually a certain path between the color area and the grapheme area, which goes before it goes to the multisensory nexus. These two areas are activated. So that would be a proof for the cross-activation theory. For the associators, she found something different. She found that first the graphemes are processed, then it goes to the multisensory nexus, and then it goes back by disinhibited feedback probably to the color area. So that's a very nice study to show, okay, we have to keep in mind that all synesthetes are not the same, and we have to try to find these groups if possible. This might be evidence that we can kind of do it. Okay, let me conclude for the first part. 2 minutes? Oh, that would be difficult. I'm not going to conclude then. Multisensory integration. Multisensory integration has not been studied so much. So this is all unisensory processing, and it showed that additional areas could be activated. But we don't know how that works in synesthetes yet. So we have the different areas again. You know them already, so I'm not going to repeat myself. And we're going to look at one experiment, and that's the double flash illusion. You show two bleeps, and one flash. And what people perceive is, also non-synesthetes, either one flash, they're supposed to report the flashes, or two. And when they perceive two, even though one was only presented, that's the illusion. And if they perceive that, it's more multisensory integration. Okay? And I'm going to skip that, all of that. There are two studies with synesthetes on the behavioral level, no MEG or fMRI, and they showed that, one study showed that synesthetes are actually having larger multisensory integration. Yeah? So they are better at integrating different senses. Another study from Hanover showed they are worse in comparison to controls. So Our goal is to look at this again and either replicate this study or this study, and in addition to that, look also at MEG data. These are the first results. We're just going to look at the illusion. This is the illusion condition, and the orange part is the amount of the illusion. We had two different types of synesthetes. That's the grapheme color synesthetes and the audio-visual synesthetes, and of course the control subjects who don't have synesthesia. And what we see up to now, but this is not a complete sample yet, is that there's not a very big difference between those subjects. The question is, why is that? And the question is also, can we find, despite the fact that we didn't find any behavioral differences like the other two studies did, will we be able to find differences in brain activations and how the different brain areas are actually syncing or not syncing? I'm quite positive about that, and I hope we will find it. But we'll see. So maybe in 1 year, I will have results to that. Okay, because of the time, I'm going to stop here. I would like to thank my colleagues in Hamburg and all of my cooperation partners and the funding from the European Research Council, and of course all of my participants and all of you for listening to me and for your attention. Thank you.
Thank you very much, Sina, for this intense insight of very current research, of an ongoing research. And I think it can be really interesting now, after the foil of artistic research with, on synesthesia, now neurological research on synesthesia. And I find it intriguing after the earlier presentation to think about a radical classification, an analytical pure definition, and the way of research. Basically, maybe later on in our discussion we can actually combine you two. But maybe first to the audience, do you have questions to or comments? Yes, please.
Yes, I have a question. You mentioned you always need inducers, something to induce synesthesia. What happens when we are asleep? For example, sound color synesthetes, if they hear something while they are dreaming, do they see the colors?
That is a very interesting question. Honestly, I've never thought about that. I think you would have to ask a synesthete who can maybe answer that question, because I don't know.
You never did MRI scans with sleeping synesthetes?
No.
Okay.
I don't know of any studies. I don't know whether, no. Very good idea, though.
Okay.
Yeah.
We have a question here, Birgit.
Yes, I would like to pose the cultural question once again. I'm very fascinated that it's so much about the alphabet graphemes, and I would like to know what is scientifically known about this connection between the alphabet, which is a very certain notational system. It's already optophonetic, you could say, and it's a very abstract mode. And what is the answer to this? Is it really like only in school people learn when they are trained in writing, trained in a cultural technique, so to say, that there is a connection between this? And what, for example, in difference to other notational systems like Chinese, of course, what happens here?
Yeah, that's very difficult to answer because, you know, especially with the graphemes, we learn that at school. So what happens actually before that? We don't know. There's very little research about children. Developmental research has to be done, definitely, because we don't know what happens before you actually learn these things. Yeah, I just recently measured a kid from my colleague who had weekdays and colors. And it will be very interesting to see whether this child will actually develop also color grapheme synesthesia. With Chinese symbols, for example, if you show a color grapheme synesthete from Germany or from England or from wherever, these symbols, you probably will not have colors. The question is actually, how is it wired in the brain? We don't know yet. I mean, that's all.
This would be a linguistic question, so to say. What happens when people are reading and writing and doing processing this cultural technique?
Yeah.
And this would be really interesting for me to learn.
Yeah. And I think that people in Frankfurt are currently also looking into that subject a little deeper.
Okay, there was an earlier question. Yes, please, and then you will come.
Thank you. So I have a number of questions, and I'll see in which order. Oh, to get closer, okay. So first of all, just also to mention something about letters, culture, sort of the cultural processing of letters. I actually saw a talk by David Eagleman in 2010 who said that potentially there is a relationship between the shapes of the numbers and the shapes of the letters, and this is what sort of connects, is responsible for the color that different letters and different different numbers have. Let's say what looks like 8? I don't know. Okay, now I'm just trying to sort of imagine a way like. 4 looks okay. Sorry, I can come up with an example, but it's there was sort of this idea that potentially the shapes are what the shapes of a letter and the shapes of a number is what determines it, rather than the cultural. So it's again more in terms of visuality than in terms of culture. But I have actually another question in terms of the projector and the associator. So Dept keeps saying that she sees something in her mind's eye, but on the other hand, she says, well, 7 is on this side here, let's say on the left, and maybe it's on the right, but she will be able to correct me for sure whether 7 is on the left or on the right. And this to me seems like a projection. And also the calendar which is around her waist, it also seems like a projection. So I'm sort of wondering whether maybe she's an associator when it comes to the grapheme color synesthesia, and she sees that in her mind's eye, and maybe in terms of the other types of synesthesia, she may be a projector. And also in terms of gaps, so she says, okay, the whole thing with space and numbers, it doesn't quite work because I have only one, but I am aware that there are gaps in many synesthetes sort of fields of perception. So I was wondering what you could say about that, or what Dept and you can say about that.
Yeah, the projector and associator distinction is just one possibility to disentangle, especially grapheme color synesthetes, I would say. Because with that, it's very easy to ask people how they perceive it, whether it's in the mind's eye or projected onto a screen. With these spatial concepts, it is a little bit different because we are used to point to things in the room, into our interpersonal space. So I would probably not make the distinction there because yeah.
We had a question here, please.
Well, it might be a stupid question, but I dare it. No, just to know, in when you are asking people, I mean, what is the proportion, if there is any, of people who answer that consciously there is no any link for them between a letter and a color?
You mean the ratio between synesthetes and non-synesthetes, or?
People who say they don't see any color.
Well, the thing is that
connected to a letter. I mean, consciously. Yeah.
Consciously. Well,
it means.
Yeah. It is quite difficult to say. Is synesthesia, is it conscious? It is made conscious when we talk about it, I would say, right? But you mean the people who say they don't see any color?
Yeah.
Those would be the non-synesthetes, right?
Yeah. What is the proportion of them?
Well, the point is that we try to cooperate with people who can give us already addresses from synesthetes. So I cannot really say. I'm not doing a study over all Germany or all Hamburg to be able to say the ratio. What's written in the literature, there's a wide variety between 1 to 25,000 people and 1 to 2,000 people who actually have some kind of synesthesia. And it differs among the different forms. For colors, grapheme color synesthesia is much more often than audiovisual or, for example, personality and color synesthesia. Did that answer your question?
Not really.
Well, then you come to me afterwards, okay?
So we had Dept and Agnieszka and then Romy.
Thanks for a great talk. I was very impressed about this study. I don't know what the name of the woman was, but about projector and
Yes.
associated. I
Tessa,
think that's
yeah.
kind of groundbreaking, no? In the
yeah.
field?
Yeah.
I haven't seen this study before.
I can send it to you if you want.
Yeah, thanks. I also just wanted to hear a little bit about where you would like, how you would like to use this research. If you know, like, if aside from classifying synesthesia and be a part of our club, like, then how, where else would it lead? Like, will it possibly lead to studies on schizophrenia or like more interesting things actually, like more crucial or more where we need more research?
Yeah. Well, I hope to find something that gives a hint that synesthesia is a developmental phenomenon. And thus we could study neural development per se, like basic science, you know. I have first of all, I have an intrinsic motivation to study that because I want to know, are they activated simultaneously or not? But then again, if we have these results, we can also understand, like I don't like to say normal, but non-synesthetic multisensory integration, because we already know a lot, but we don't know everything. And this could lead to a better understanding in non-synesthetic multisensory integration, but also, like you said, schizophrenia is a very interesting phenomenon, and I know that, like for example, Tessa is already working on that as well, where she has different hypotheses, also looking, comparing actually synesthesia, schizophrenia, and other psychiatric disorders. Can we learn from synesthesia how to make other disorders better, for example? But that's very complex, and I'm not very into it, but that would be quite interesting to look at, definitely in the future.
I would say time-wise, two last questions or comments, Agnieszka and Romy.
Yeah, I just have two comments, really. The first one is actually regarding this idea of the link between learning graphemes and synesthesia. I believe Richard Sotowick, from the neurologist from America, said that assuming that there is a gene for synesthesia, it seems that in majority of synesthetes, it's expressed at the time when they learn alphabet. But there are people with synesthesia who don't have their synesthetic experience in response to graphemes. They only have it to the sound of the phonemes. So he suggested that in this case, this gene for synesthesia is expressed earlier. So I think you do have people who have synesthesia just for phonemes. So it seems to be linked with graphemes, but it's not necessary. And also another comment is just regarding the study that you mentioned by Hubbard and Ramachandran on the visual search task. So correct me if I'm wrong, but I don't think this study has been replicated, and that this is the only study who found that this pop-out effect. And then later on, I think Noam Sagiv said that, in fact, you need attention to get those synesthetic experiences. So they might be automatic and they might be involuntary, but if you sort of overload synesthetes with some other task, it actually doesn't happen. So you need attention to experience synesthesia.
Yes. Yeah, I didn't mention those studies, but yeah, they have shown that you need a little bit of attention to actually have this synesthetic perception. That's, right. Thank you.
Okay, so I actually wish to draw on points that were raised here before about the optophonetics and the spatial relation of, let's say, both reading and color and even the sounds of words. So I'm addressing my question to both you, Sina, and Dept. And I was wondering whether there are different ratios within cultures, be they Asian or from the Middle East, or cultures that write and use different kind of phonetics and different kind of graphemes, and whether we know if this association of colors and even sounds that accompanying graphemes are divided differently in different regions?
Yeah. Good question. Again, I cannot answer that. I don't know of any studies who have looked at Asian populations or African populations. I'm not aware of them, but it would be very interesting to look at that, definitely.
What I found, it seems that it's the same somehow. Like, you know, it's like for Chinese characters, there are like I think there's a Japanese Synesthesia association, and there's like an Australian, and so I haven't noticed that it's like very different. It's just characters. It's just the science again. So then, one Chinese character has one color or one taste or something, and then. Yeah. That's interesting. I have not done any research on that, and but I would be as brave to say I don't think there's a difference. But it's just how I've experienced this whole research, that it's like you hope that there's some specific about philosophy or how you perceive the world or culture and stuff, but then as deeper you go into it, it's just always resolving itself, because then you find a new person who is then has a linear time experience or something. So, but it would be interesting to go like Asia, India, or like in terms of religion also, and especially in terms of space and time, which are completely different ways of sensation, you know, if it's even a sensation. Like for me as a synesthete, you know, if I speak to a Japanese or Chinese person, it's like, or like as I said, if it's a Finnish, then I see the colors, but it's just me that is that are like putting my European alphabet onto their sounds. So that's the first translation. And when they write down their names or something, and I see it on paper, maybe the colors disappear because then it's not that anymore, because then it's different. You know what I mean? Or so it's about visualizing language.
Okay, I would just say let's come to an end of our first day 2 panels. Thank you so much for the presentations of Sina and Dept. I think the conversation will go on. We just opened up the whole field.